on characterization of virus infecting capsicum enum in Eastern UP. Today, my topic is a sign on the molecular characterization of viruses, especially in plant viruses. Am I audible, everyone? Please, ma'am, am I audible?
Yeah, yes, you are. Yes, you are audible. Yeah.
So, especially, I'm working on the identification of viruses here in the lab of crop improvement lab, and my senior scientist is Dr. Achyut Kumar Singh, is working on the CRISPR-Cas genome editing crops. He has 36 crores projects sanctioned by ICAR, in which there are 6 to 7 vegetable crops which are going to edit gene on the basis of uh, 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 abiotic stress as well as biotic stress suffering from the crops. So especially I have little bit contribution in it. Uh, so this contribution in the project I have done, especially on the viruses, vegomoviruses, which is very harmful to chili crops as well as tomato crops. So I will characterization of molecular levels, which vegomoviruses. So we are uh, going to express my works through these webinars, especially when we are going to characterization of molecular level of viruses. First of all, we going to collect the sample of different local areas, and then we are going to preserve the samples. First step of molecular characterization was done from the extraction of DNA from the plants. We are going to use to CTAB method and extract the plant's DNA, infected plant DNA. And then second step, we are going to follow the PCR. PCR having a polymerase chain reaction, the DNA which is infected of the viruses, we are going to uh, uh, generate a multiple copy of the viruses DNA as well as the plants DNA. But when we perform the PCR with specific primers, then we going to desired of the gene which is multiply in PCR. When we are performing the viruses sample, we have selected two types of primers, that is beta primers as well as Rojas primers. These two primers, which is especially we are used in the amplification of plant viruses. I think my laptop are not properly works. That's why I'm not going to represent the slides on it. But we shows some photos are available on my laptops. Through I show you with back cameras. Uh, Once again, this is my works, and which are especially I'm going to today conclude the introduction and as well as survey of virus infecting and preservation. In this slide, we are introduction. Plants viruses are viruses that affect plants pathogenic to higher plant, especially the plants viruses are going to infect the higher plants, and reduce the yield qualities of crops, that is chili crops as well as in tomato crops. We are in working in India as well as in eastern UP. So this is the some surveys was done by me. This is the healthy plants, and this is the infected plants, which is viruses. Some other surveys I have done. This is the white plants as well as this is the infected plants, and this is the hybrid plants. And this is the slide of uh, extraction of DNA, and we use some precaution when we are going to extract the DNA, because DNA are going to multiply, and when the contamination of DNA, the whole experiment was not performed by well. So we are very precautious measure to take by me in the extraction of DNA. Then we perform a PCR. A PCR, this is the machine in which we are going to multiply the desired gene, yeah, desired gene of sequence, in which we are use the different PCR components like DNA samples, 10x buffer, and MgCl2, DNTP, and primer. There are two type of primers we are using PCR. First of all, reverse primer as well as and forward primers, and then we use tag polymerase. This is used for the stability of DNA when we are going to amplify the DNA. First of all, denature the DNA at 94 degrees Celsius for 4 minutes. Then primer initiation of DNA, which is 94 degree for 1 minute. And this is the first annealing temperature, which is done by the 52 degrees Celsius for 45 seconds. Then second annealing temperature is 72 degrees Celsius for 1.50 minutes. And second step, which are involved in 24 cycles. And then rest of temperature, which is set up in the gel the PCR. When we complete the PCR, we are performing the check of DNA quality. The DNA quality check by the gel electrophoresis. When we are going to check the DNA quality, we make the 0.8% gel, uh, and when we are going to load the check the quality of DNA, we are going to load the some I think 2 microliters of DNA, and 8 microliters of distilled water, and 2 microliters are dye, which are going to mix, and then load the sample in gel when the gel costs, and then run the gel on the 65 volt, and this is the gel images which are performing when the performing uh, this is the images shows like this. And when the band is clear, this DNA purification is very good in amounts. When the PCR perform after, then we have find uh, desired DNA sequence. Then take the DNA sequence and go through the rolling circle of amplification, because the viral DNA is circular in nature. So we can't perform a PCR, because PCR having a single circular strand DNA, which are used. But when we are going to viral DNA amplify, we are not going to do the PCR. we are going to do a special type of methods which are known as rolling circle of amplification. In rolling circle of amplification, we have simple buffers, DNA templates, and those are mixed together, and which put on the PCR machine where 95 degrees Celsius for 4 minutes, and then remove the PCR tubes and pull down the PCR tube, and then add master mix, and this sample are incubated 18 hours for 32 degrees Celsius. When the PCR is complete, when the rolling circle amplification are complete, then we are performing some special type of experiments called as ligation. When we are going to ligation, we have a desired gene fragments which are going to clone. That is uh, uh, I have two types of gene of interest.
Yeah.
There are 2.7 kb and 1.3 kb. The Bergamo virus, they have DNA A and DNA B, which is 2.7 kb of genes, and beta satellites have 1.3 kb
Yeah.
of genes. Then we perform the ligation. We use PKS vector. PKS vector, I especially use the PKS vector, which is 2.9 kb in the gene size. Uh, and
ये
when
PhD
we are going to
ये वर्क है क्या?
perform, when we are going to perform,
हाँ
this is the blue uh, P blues skip vectors
ये तो
images.
MG कर रहे थे तो
This is the
ये क्या हो गया? सर्किल पाया सर्किल ओके पैरामीटर होता है या रेडियस मैं भूल गया.
Hello. By mistake, you are muted. Please unmute yourself. Please unmute.
okay, sorry, sir. I am use the PKS vector in this experiment. The PKS vector is 2.9 kb size. In PKS vector, there are ampicillin resistant. So we are going to use select the colony. We are use the ampicillin. So this is the ampicillin resistant gene and lax Z gene. When we are going to insert that gene of interest in PKS vector, we have different size of restriction digestion enzymes. So we use the BAMH1 enzyme for the digestion of that vector, and as well as my gene of interest. When we are going to perform a ligation, we have same enzyme to digest vector as well as gene of interest. When we achieve digested PKS vector as well as gene of interest, so then perform the ligation, because we have not done the same digestion of PKS vector as well as gene of interest, the ligation is not going to complete in that way. So we have digested PKS vector as well as digested gene of interest. Then we are going to do the ligation. In ligation, we use the RCA insert in rolling circle amplification insert, which are going to digested with BAMH1, and PKS also digested with BAMH1, and use a buffer, ATP, and T4 DNA ligase. When we are going to perform the ligation, we use the ligation enzyme is known as T4 DNA ligase, and some amount of water, that is MQ water, milli-Q water, total reaction which are in 20 microliters. In which RCA insert is 8 microliter and PKS vector is 4 microliter, buffer 2 microliter, ATP 0.5 microliter, and T4 DNA ligase is 0.5 microliter, and rest 5 microliter are water is used in the ligation. When we have going to ligate this vector, the ligation was complete in 16-hour incubation. After 16-hour incubation at 16 degrees Celsius, the ligation is complete. When all things are mixed together, we are going to incubate 16 degrees Celsius at 16 hours. After this, we are going to store this reaction in -20, and then after utilize that. So when the ligation product are achieved, we are going to perform transformation. Transformation especially happen. We have to use DH5 alpha competent cell. Everyone known. Can you tell about the DH5 alpha competent cell here? Hello, am I audible? Hello.
Yes, sir, you are audible.
So I'm going to talk about the DH5 alpha. This is the E. coli E. coli variants which are used in transformation. DH5 alpha have capability to transform gene of interest with vector. So DH5 alpha, we are having DH5 alpha always in -80. So we take DH5 alpha. DH5 alpha is 15 microliter as well as in 100 microliter. We have going to store in the -80. This is the 15 microliter are in 100 microliter. So we take DH5 alpha from the -80 and thaw on the ice, as well as we also ligated product on the ice, because ligated product are also put in -20 degrees Celsius. So this is some flow charts. So the ligation the transformation was done with the help of DH5 alpha and ligated product. We are going to use two type of gene fragment. First of all, already I told you there are two type of gene interest. That is 2.7 kb, that is DNA A and DNA B of Bergamovirus, and second gene of interest is beta satellite, which is have 1.3 gene kb size.
Yeah.
So we have two types of gene interest, that is 2.7 kb as well as 1.3 kb gene of interest. So this experiment was performed by two type of gene interest. So we are going to use a DH5 alpha competent cell and RCA ligated vector in totally pour the RCA ligated sample in DH5 alpha in laminar air flow, because some contamination is occur when we are going to open lab atmosphere. So we have done all the experiment in laminar air flow. Already I told you this is both sample are going to thaw on the ice, and after thawing 14 minutes, we are going to give the heat shock 42 degrees Celsius for 2 minutes. Then some other step are taken by me. Then incubate our eyes are also 10 to 15 minutes. Then we add 1 ml LB in it for the growth of DH5 alpha with ligated insert. After adding 1 ml LB in it, it incubate on incubator at 37 degrees Celsius for 1 hour. So when the complete 1 hours, we take this sample, then incubate, then centrifuge at 12,000 RPM for 4 degrees Celsius and 5 minutes. And then some other precaution which is taken by me. And the lastly, we have found the plasmid of that transformation product, and then we are going to next experiment to perform by me, blue white screening. When the going to blue white screening, why the blue white screening is important? Because when the transformation occurs, other bacteria are not transformed bacteria are also growth in this culture. So this is why we are going to blue white screening. Where I'm already told about the flu PKS vector have lack jet gene. Those vector have lac jet gene, and then these lac jet gene sites also have the MCH region, meta cloning site region, in which different digestive enzyme sites. So BAMH1 going to cut that lac jet gene sites. The lac jet gene are not going to express. So on the basis of these, we are going to perform the blue white screening. In blue white screening, there are 3 types of chemical we are used. First of all, IPTG and XGAL, and then ampicillin. Because the vector which also have ampicillin resistant gene, so when the transform these vectors, the only those vector having bacteria are grow, they have the ampicillin resistant gene. And transformation was checked by the IPTG and XGAL. When the IPTG and XGAL are used in the LB, in the LA, in LA, these 3 things which are going to mix in LA in available in the lab in the solid form, which are first go to melt the LA and add 200 microliter XGAL and 100 microliter IPTG and 100 microliter ampicillin when are going to the blue white screening and mix well. After mix well, we are going to make a plates, petri plates of this LA medium, and these LA mediums which are used to blue white screening. The transform plasmid which are going to smear on it, and on the basis of blue white screening, we are going to observe which bacterial cell are transformed or not transformed. Then we are going to incubate 37 degrees Celsius for 16 minutes. After next morning, we are going to observe that petri plates, the blue colony shows the not transform, and the white colony shows the transform of gene of interest. When we are going to transform gene of interest, we are going to pick the white colonies and then perform the different digestion with that colonies. That colonies were also going um, to isolate the plasmids, and plasmid are dissolved in 30 or 25 microliters, and then we took 4 microliters from these plasmids, and we use uh, um, different BAM-H1 enzymes. BAM-H1 we have especially use there are 5 type enzymes: BAM-H1, XBA, EcoR1, and KPN and PIST1. There are 5 enzymes which are used in the transformation check by me, and I show some slide on my laptops, which is have uh, 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 some was, images of these enzymes. Uh, how can separate different bands. Especially, I'm not able to show that things. I've told orally. When we are using a digestion check of these transform plasmids, we are going to cut the different size of bands. I think these are the very crucial process in which we have to depth knowledge of enzyme sites, as well as vector size, and where the enzyme are going to cut in vector sequence. So when the digest the plasmid with different type of enzymes, we uh, we uh, we uh, 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 we use first BAM-H1 enzyme, because we are cut the vector as well as gene of interest with BAM-H1 enzymes. So when the digestion is done, the 2.7 kb fragment will appear on this gel, as well as 1.3 kb of band size will appear on this gel, and the vector size are also appear in this gel. Some non-ligated vector are also in it. So there are multiple fragments are in the gel images. First of all, my gene of interest is 2.7 kV. So in one plasmid, there are 4 bands are appear: 1.7 kV as well as 2.3 kV. These are the both gene of interest, and 2.9 kV gene are bands are also shown in that. This is the PKS vectors. So these confirmation test are happen by me in this experiments. When the gene of interest are totally find in transformation, we are going to plasmid isolation, because without plasmid isolation, we are not going to sequencing. In other way also going to sequence that plasmid, but these way is very helpful to find out the real sequence of any pathogen. So first you go to DNA isolation, then go to the primers, and then primers amplify your gene of sequence, and then you are going to RCA, and RCA product was digested with BAM-H1, and those vector which are used are also digested with BAM-H1, then going to ligation, then going to transformation, and transformation, after transformation we are going to blue white screening, and then blue white screening we are going to plasmid isolation, and after this we found the gene of interest of the plasmids, and these plasmid are sending to the sequencing and the phylogenetic analysis of these viruses. these work are done by me in IIVR labs, Indian Institute of Labs, and there are some scientists which are help in my research works. The renowned scientist is Dr. Achyut Kumar Sen, which is also working on the uh, abiotic and biotic stress. And some other scientists are Dr. Endeavor Prasad, which is especially working on chili, because my research crop are chili. So some suggestions are also taken by me from the Endeavor Prasad sir, and one of the renowned scientists of biotechs here, Dr. Suhas Kurkute, is also suggesting me the vectors, which vectors are beneficial for me, and as well as which enzyme are going to digestion and transformation. So these are the my PhD work in IIVR. I have some publications in review as well as in papers. Top 10 viruses infected on chili in Southeast Asia. This is my review articles, and I was selected for Young Scientist Conference in 2024 in RCB Faridabad, and I have also attended the Indian Citrus Congress in Nagpur, and one thing I tell you, Dr. Deshmukh sir was I'm meet in September 2023. The sir was coming in when the MOU was done with IIVR and my universities, Deen Dayal Upadhyay, Gorakhpur universities, where I'm going to receive the sir and make the conversation with sir. Sir are very polite and very humble. So very thankful to sir and all the member here. I'm very thankful to inviting me as the lecturer of PhD research works. This is my works. So, ma'am, this is from my sides. And thanks again for the all members. Hello, hello.
Yes, yes, you are audible. Yeah.
Yeah, ma'am. This is my complete journey of research in IIVR and Deen Dayal Upadhyay, Gorakhpur University, Gorakhpur.
Okay, thank you. If Deshmukh sir is present here, please, sir, if you could wind up for the session.
Hello.
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 Yes, I think sir is there.
Yes,
Hello, sir.
yes.
Yes, yes,
Hello,
sir.
sir. Pranam. Myself, Jitendra Kumar Kushwaha, and I'm reintroduced with you, sir. I'm especially first time meet with you, sir, in Deen Dayal Upadhyay, Gorakhpur University, and I am received in September, where the MOU is done with IIVR. The director of IIVR is Dr. T.K. Behra is also there, and sir is also there. So sir is very humble and down to earth. I meet especially in that times. So sir, please.
Yes. So today, really, I congratulate Malini ma'am as well as yourself.
Thank you, sir.
The
Thank you.
reason is that Malini ma'am is continuously arranging various lectures and every Sunday. And your explanation I have attended, your research presentation I have attended, It was a really a nice, so to appreciate your efforts.
Thank you, sir.
Yes, I want to appreciate, and really you are doing a very well. All the best to you in the future. Now,
Thank you.
there are certain questions in the chat box, and other people may ask the question.
Yes, sir.
I request Malini ma'am, go ahead.
Yes, sir. Thank you, sir, for your nice appreciation. So I would ask the participants if you can unmute yourself one by one and ask directly, or rather the speaker, you please, if you can go answer the questions one by one.
Okay, okay, ma'am.
There are few questions in the chat box. Madam, there
Yes,
are
sir.
questions in the chat box.
Okay, then I'll read out the questions then. Yeah.
Okay, ma'am. I'm unable to look.
Okay.
So,
Here is it is written by Kanvi Madam, I think. Digested DNA fragment means, and second question is LA medium means, and third is which sequencing technique was used.
okay, ma'am. Okay. The LA in Luria agar, which are used in the plating or growth of the virus bacteria in my labs. The LA is the medium where the growth of bacteria. And second one, the second question, ma'am.
Is DNA fragment means.
DNA fragments. Okay, ma'am. When the digestion of the uh, viruses, the viruses is double standard viruses. In Vagmo viruses are double standard viruses. So the digestion of DNA means we are going to we have to digest with BAM H1. When the digestion of BAM H1 of the fragment size of viruses, it happen in this experiments. The viruses are digested with BAM H1 as well as vector is digested with BAM H1. The both are going to ligated. We are not going to ligate digested with BAM H1. The vector was not going to take their gene of interest with ligation. So ligation require the digestion of the same enzyme with gene of interest or DNA of the vagomovirus as well as the vectors. So this is the digestion of DNA of the vagomoviruses. And sequencing, we have to try to go to sequence in DU University, Delhi University. So I don't have proper name of the sequencer because the sequencing was done with my principal scientist, Dr. Achut Kumar Singh, which is take that forward action on it. So they have the data on the which type of sequencing is done in the case of this experiment. So I have little bit knowledge on it. So I don't go through that. So, ma'am, next question.
One
Please.
more question is there, that is transformation check. I think she's asking.
I think the very important process transformation check was done by two things. First of all, blue white screening, in which only going to observe which cell are grow. Superficial check, the transformation was done by the blue-white screening, but we are really confirm the gene of interest are transformed. The transformation plasmid was digested with this enzyme which is happen before we are going to ligation. Ligation में जो एंजाइम वो डाइजेशन एंजाइम यूज किए थे, वही एंजाइम से आप जो है ट्रांसफॉर्मेशन चेक करते हैं. इट मीन्स when the ligation was done, this enzyme is used in the transformation check. That is, I'm use the BAM-H1, so I'm use the BAM-H1 after the transformation. We found the same type of band before the ligation. Before the ligation, I have two type of band size, that is 2.7 kb and 1.3 kb, and the vector size is 2.9 kbs. So after the transformation, we are going to digestion, we found the same type of DNA fragment on gel. It confirmed the ligation is occur, and this is the way to check the transformation. This is done by me. Okay, ma'am, this is the answer of the transformation checked by the digestion. Hello?
Yeah, thank you so much.
Okay, ma'am.
We have Prakash Alami sir also with us. Sir, if you would like to interact, so and after that only we'll conclude then.
Not exactly anything. He is trying his best to complete his work. So let us wish him a good luck. ठीक है ना जितेंद्र, अभी तुम्हारा तो PhD चल रहा है ना?
जी सर,
जारी
अभी
रखो, अच्छा काम करो.
हाँ सर, चल रहा है, और इसके बाद हम लोग वो
सीक्वेंसिंग के लिए भेजना है.
सीक्वेंसिंग के लिए प्रोडक्ट रेडी है, उसको सेंड भी कर दिया है, और इसके बाद इन्फेक्शियस क्लोन हम लोग बनाने की कोशिश कर रहे हैं, इसमें पी कैम्बिया वेक्टर का यूज करके.
ठीक है.
Thank you, sir. And next plan I am working on the especially on a resistant gene of against the viruses, because a lot of scientists work in, and I read the paper, one of the paper of Dr. Mahendra Prasad, NIV PGR TOLC resistant gene. So I want to do the PDF on find out the resistant gene against the viruses.
Okay, so thank you. Yes, thank you both to our speaker for today and all the participants. So we will meet once again, hopefully in the coming week. So till then, bye, and have a good Sunday. Okay.
Thank you, ma'am.